Hey everyone! In the last video you learned how to count chromosomes, DNA molecules, and gene copies as a cell proceeds through cell division. Now in the next two modules of this chapter we'll examine meiosis and mitosis in detail. Before we do, I'll spend this video briefly summarizing these two processes and we'll discuss their importance as well. We have two questions this time. And our two questions are: what cells are produced by mitosis and meiosis, and how do these cells differ? And how do mitosis and meiosis function in the growth and reproduction of organisms? In eukaryotes, an organism's cells can be categorized by whether that cell's chromosomes and the alleles on those chromosomes will be passed on to the offspring of that organism. Diploid somatic cells, or human body cells like these skin cells, pass on their chromosomes to other body cells, but not to offspring. The prefix soma means body in Greek. Haploid gametic cells, or gametes, do pass on their chromosomes to the offspring. In humans, somatic cells and gametic cells are produced through different types of cell division. Once again, we'll follow chromosomes 1 and 20 and the hypothetical genes A, B, and C to illustrate how human somatic cells and gametes are made. For the production of both gametes and somatic cells, the cells that will produce them first replicate their chromosomes. In a diploid species like humans, after a cell replicates its chromosomes, it now has 4 copies of each gene. After these diploid somatic cells replicate their chromosomes, they divide through mitosis, which is a method of cell division where one parent cell produces 2 daughter cells with the same number of gene copies, in this case 2, that the parent cell started with. These daughter cells have the same genotype as each other and the same genotype as the parent cell, assuming no mutations occur. The parent cell has essentially cloned itself through mitosis. Since only one cell is required for mitosis, this is a form of asexual reproduction. Gametes, on the other hand, are produced in different ways in different species. In animals, gametes are generally produced directly by meiosis. Unlike mitosis, meiosis involves 2 cell divisions, meiosis I and meiosis II, with the purpose of producing haploid or 1N gametes that have only 1 copy of each gene. After DNA replication, meiosis I produces 2 daughter cells, distributing 2 of the 4 copies of each gene into each of them. However, unlike in mitosis, the genotypes of these 2 cells are different from the parent cell, because of processes called crossing over and independent assortment, which we'll cover later in the course. Both of these cells divide again during meiosis II, but this time without going through DNA replication first. So each daughter cell therefore only gets 1 chromosome of each type and only 1 allele of each gene. These 4 cells are haploid and develop into sperm cells in males, and all 4 will have different genotypes. Meiosis in females is similar, but only 1 gamete, the haploid egg, is produced. As we mentioned before, the sperm and egg fuse during fertilization to form the diploid zygote, which gets 1 allele of each gene from the sperm and 1 allele of each gene from the egg. that single-celled zygote replicates over and over by mitosis to produce the multicellular organism. So we can see that meiosis, fertilization, and mitosis are all needed to produce the offspring. Next, we'll examine the functions of meiosis and mitosis. The function of meiosis is to produce the gametes with different genotypes. During reproduction in humans, a 1N sperm with 23 chromosomes and a 1N egg with 23 chromosomes fuse to form a 2N equals 46 zygote. So the genetic function of the gametes is to each pass on one set of chromosomes to the zygote, which will eventually form the offspring. In this zygote's karyotype, 23 chromosomes came from the sperm and are termed the paternal chromosomes. The alleles on those chromosomes are called the paternal alleles. The 23 chromosomes from the egg are the maternal chromosomes, which carry the maternal alleles. At this point, the offspring consists of only one cell, the zygote. Here's a real zygote, and you can see the nuclei that came from the sperm and the egg. As we saw before, the zygote then undergoes many rounds of mitosis to produce an adult. So mitosis builds a multicellular organism from a single cell, the zygote. Nearly all the cells in your body were produced by mitosis, which means that nearly all of them are genetically identical, with the same chromosomes, the same genes, and the same genotype, assuming, of course, that no mutations have occurred. During an organism's lifetime, somatic cells die. A second function of mitosis is to replace these cells. Human somatic cells can divide about 50 times, and then can no longer reproduce, though this number depends on the cell type. The number of times a cell line can reproduce is called the Hayflick limit, and it's different for different species. This is one reason humans are not immortal, and this might contribute to the decrease in cell functionality that accompanies aging. One human disease caused by mutations that alter the structure of the nucleus, thus causing problems during mitosis, is progeria. Symptoms include those normally associated with aging, like loss of eyesight and cardiovascular problems. But in progeria, they present in young people. This is reflected in the etymology of this word. The word progeria comes from the Greek words pro, meaning before, and geros, not sure if I'm pronouncing that right, meaning old age. Study of this disease and the mutations that cause it may lead to a better understanding of the process of aging itself. Progeria underscores the importance of cell replacement by mitosis. In fact, during the time you've watched this entire video, millions of cells within your body will have died and been replaced by mitosis. A third function of mitosis, important in plants and fungi, is to produce haploid cells from other haploid cells. Following the initial production of haploid cells by meiosis, in plants and fungi, these haploid cells undergo mitosis, producing more haploid cells, only a few of which end up as gametes. In plants, this production of haploid cells from other haploid cells through mitosis is responsible for the production of gametophytes. These are the multicellular haploid structures that house the sperm and the egg. In fungi, single-celled haploid spores undergo mitosis to produce haploid multicellular hyphae. So we can see that in plants and fungi, meiosis and mitosis work together to make the multicellular haploid structures that form part of their life cycle. Mitosis also plays a role in producing plant gametes, but we'll cover this in a later video. So mitosis is important for building organisms, replacing cells. Without mitosis, we'd all still be a single cell. Thanks to mitosis, humans have around 30 trillion cells in our bodies. Okay, so at this point you should be able to describe the types of cells that are produced by meiosis and mitosis, as well as the importance of these two types of cell division in reproduction and development. If you don't feel like you can, as always, feel free to watch the video again. And that brings us to the end of Module 1 of Chapter 2. In the next module of Chapter 2, we'll examine the cell cycle and mitosis in detail. See you then.